एक पूरा डब्बा तब पर राजस्थान जीत गए लास्ट बॉल में जो बाउंड्री मारा और आदित्य ने बॉलर इसलिए मुंबई का इतना जीत गया ठीक है तो कुछ बोले मत अरे मैच है क्या मैच है तू क्या बोल रहा है अरे यार ऐसे नहीं फुटबॉल खेल रहे हो जिसको पंजाब अरे पंजाब ने डीसी टेस्ट किया तो डीसी वाला पंजाब वाला पंजाब वाला हमें डीसी टेस्ट क्या है अरे पंजाब आ रहा है एक मालूम है क्या मैच है ए पंजाब आ की मैच ना डीसी टेस्ट पंजाब आ की मैच है क्या भाई दे एक अगर डीसी टेस्ट करेगा मैं तेरे को आके मारूंगा मिसिस मारेगा तेरे को क्या पंजाब आ रहा है तो पड़ी मिसिस को मार दो क्या उल्टा हो गया ना अभी पंजाब आ रहा है मार ले मिसिस को अच्छा ये ना कुछ ज्यादा ही लिया पहले मैं उसको मुक्का है तू हां आज के बाद कुछ फर्क नहीं पड़ेगा आज के बाद तो मुझसे मिलना है वो घंटा पर भी मेरा को दिन का इसी और पंजाब का भाई मुझे खा ले दोनों भाई ऐसा क्या कोई नहीं दिया भाई ऊपर चाय ना खेल क्यों रहे मुझे काय ना काय प्रश्न ऐसे फॉर्मेलिटी असते प्रत्येक टीमला 14 गेम्स खेळायचे ऐसा बोले क्या है हम क्वालीफाई होते चढ़ निकल जाते हैं हां थोड़ी तो खेलना तो पड़ेगा ये क्या बोलते हैं दिल्ली तो क्वालीफाई भी नहीं होगी दिल्ली तो खेल रही है ना वो दिल्ली मचा रही है हां दिल्ली हम लोग सब क्लियर कर दिए हैं सबको लेके डूब रही है हम साथ साथ ले डूबे मुंबई की ही मार ये क्या हो रही है इसी का अपन सब ना मैं आप जैसे जितना ब्रेक क्या कुछ जड़ के बोला काय तू मुंबई से करता है तू किसके साइड है अभी उड़ा बैठा ये बता ना वो देख रहा था लग रहा है ये दिल्ली के साइड टी-शर्ट से मैंने तो तू सीएस लिखा लगा है तू भी सीएस के क्या है मैंने नहीं बोला सीएस मैं बोला था सीएस के रोहित का फॉर्म था रोहित का मैच जीते हां जो मैच है ना रोहित का मैच हैदराबाद के साथ है क्या बोल रहे हैं वो तो जीतने पड़े हैं बहुत मजा है हैदराबाद पूरा मारे आगे लास्ट हैदराबाद में कोई नहीं खेलने वाला अरे वो हेनरी क्लासन है सेंचुरी मांगी भाई जो मारी उसको सर्विस लेकर कौन हेनरी क्लासन तो बड़ी मार गई नहीं सेंचुरी तो मार के देखा क्या वो उसका अरे कल देखा क्या हमारा वो एक ही बंदा था जो हैदराबाद से लड़ रहा था क्या इंप्रेस कर अरे हेलमेट लेकर सबसे पहले धूप लेके बोले लगा अरे वो ये उसकी वाइफ से क्या अरे मैच हो मैच के किसी को तो वीडियो कॉल किया था स्टेडियम में नहीं किया बीबी को करना लगा था नहीं ऐसे क्या फोन पे बोला फोन पे नहीं मतलब कि आई नहीं था मैच देख रहे थे थाले में ऊपर बैठी है वो नीचे है फैमिली को आने देते नीचे वो शादीशुदा आदमी है शादी क्यों है वो गर्लफ्रेंड को बोलते हैं बहुत विराट कोहली का करियर खत्म हो जाएगा अनुष्का शर्मा में निकल गई सलमान नहीं बच गई फोकस तू क्या कर रहा है उधर ओ पंकज बोला ना इसलिए मैं तू क्या कर रहा है उधर
मत लेट कर रही है
यहाँ पर लगा के
ये देवे कुछ लाया फाइनली
ये आनंद है का आनंद से
भाई
ये वो बोला है
चलिए आओ यार ये तो क्या
बोल रहे हो पूरा
वहां वेट करो
तो नॉनवेज नहीं है अब बिना नॉनवेज खाया
क्यों मैं तैयार है
तो ऐसे ही मजाक में पूछ रहे थे
सीधा
मैं
मत
तो माता को
भाई काम
मेरे सारे बीच में हाथ मेरा
सीधा मत
हां उसको नहीं आता है
भाई क्या बोल रहे हैं रोज रात को तो करता है वो फिर खाना नहीं खाता है
खाने में क्या करता
ठीक तो बाबा
खाने
खाना आप लोग खा लेता हम तो क्या बोलते हैं
ना ना तो ये पूरा कोई मूवी बैठा है उसके
बोल बोल
बोल नो कौन सा
अबे जरा हाथ में ठीक तो बाबा उनको ओए मैम जाने भी देगी
अरे हम चलो
अरे तेरी फोटो नहीं दूंगा चल तेरी फोटो नहीं दूंगा चल
मुझे फोटो लेना है
मैं डाल सकता हूं दूसरे में
ये सरप्राइज प्रैक्टिस
मेरे पिक्चर के ठीक है कौन सा चला है बीच आने आपकी पहुंचे ये तो मूवी में ये मुझे लगता है कि पहुंचे दो महीने तो बाल ही तो ऐसे बाल उठ के और बीच में उसके बाल की तारीफ कर रहा है बीच में तो मुझे तो फालतू भगवान लगता है बेचारे को हो गया क्या लग रहा था तो कैसी थी उसकी देखना सोचने के बाद बेकार थे और ऐसा खासा पेंटिंग ऐसा असाड़ होता कि पोजिशन है तेरी छोटा वाला स्माइल का टाइटल लावला है इतना हंस रही थी वो करी तो उसको रीजन था भी तरह सिलेक्ट करते हैं उसको हंसने की क्या जरूरत है चलती जा रही है मुझे कमेंट कर देना खाना कौन सा मोड़ा अच्छे से
किधर आई तारीख? अरे गांधी पॉइंट नींबू क्या नहीं देखा मुझे आती पेपर कॉल के कोर्स मैम बोलते स्ट्राइक मैम लेक्चर याद नहीं करने भाड़ा में आर्यन को घर अपना फोन ऐसे क्यों छोड़ रही है
क्या करो खा यहाँ पे ना जो हुआ
चलो
मुझे ऐसे ना वो माँ पार
क्या
अरे पानी पी रहा था तब जब मैं तो बोला जिसे ऐसे माँ पार लग रहा है उसके हाथ में
कौन बोला लेट हो गए कौन बोला ये बोला मैं कुछ बोला ही नहीं तू आ गई क्या
सुना भाई मैं भी अभी नहीं अभी नहीं
उनको अंदर ठंडे ना तेज से बैठी और बोलती आगे वाले से बात
उससे
एक मैम
तू
मुंबई आ रही है
जीत ही नहीं रही है
फैंसी जरूरत नहीं
ऐसे फेक ऐसे उसके बिना भी हम जीतेंगे और सीएसके को हराएंगे
नल्ले वाले से आगे बढ़ रहा एक बार फॉर्म आ गया
तुम लोग ये आईपीएल में नहीं खेलता
क्या बोले इंटरनेशनल में खेलते हैं
कैसे खेलते हैं
कोहली का जो फॉर्म है कोहली का सपोर्ट कर रहे हैं
बात कर रही
अगर रोहित नहीं रहता तो कोहली भी नहीं रहता
कोहली है
ये नहीं है
चल नहीं है ना बोला
अगर रोहित नहीं रहता तो कोहली भी नहीं रहता कोहली को निकालना है रोहित ने बैक किया उसको
एक्चुअली रोहित ने उसको बोला था छोड़ देता ना वो निकालने वाले थे लोग
रोहित ने बोला इसके लिए है वो टीम में
अभी फॉर्म भी आ गया
रोहित तभी आएगा
कोहली हमारा वो मैथ्स सर है ना वो सारे सब्जेक्ट
सिखाते हैं
मैं पाऊंगी चलेगा रोहित ने समझ आए थे लगा था कि हैट्रिक मिली है उसमें
रोहित ने हैट्रिक ली
हैट्रिक लिया
रोहित शर्मा
मुंबई जिला भी गया था वो हैट्रिक
रोहित बॉलर
रोहित को बॉलिंग नहीं क्या करने को मिल रहा है
रोहित को बॉलिंग
रोहित बैटिंग करते नहीं बॉलर
हैट्रिक लिया उसने
हैट्रिक लिया है हरभजन सिंह को 0 पे आउट किया नहीं वो बैट्समैन ऑलराउंडर थे अकेले ने 6 आईपीएल ट्रॉफी उठाई
एक ही बात एक दो चार के बाद
Good morning. One.
So what we were doing the other day? Absorption by root. Still wait. Go to the staff room. Rekha ma'am is there. Just tell her, ask her that I kept my marker pens where I sit on the table. Go and get those 3 pens. I just left them. Holiday is over for all of you. You are coming first time. First bio lecture, no? Second bio lecture. I never saw you.
He was wearing mask.
Huh?
He was wearing mask.
That's fine. You were wearing mask the first day. How can you come wearing mask for the first day? New place. Which school? अच्छा तुम साधु वासना नहीं कहो तुम्हारा फ्रेंड है कि उसका शक्ल देख के तुम डिसाइड करो कि पेरेंट Which topic we completed? But I think we were talking about osmosis, no? Those experiments we were talking.
Yes.
Active transport we had finished. I asked you certain questions of the book to be solved.
Yes.
Okay, so as and when we are finishing those, Selina, as and when we are finishing certain parts of the chapter, so let us complete. What did we study the other day? I must have asked you to solve 2 questions from the book. Did you?
Yes. Yes.
I had asked certain questions to be solved the other day. Moving to page 53. Looking to page 53. Look into page 53, the descriptive type D. Descriptive type D. You will try writing down these definitions in the notebook. Number 2, sub number A, B, C, D, E. This 5 first. So which are the definitions I am asking you to write? Those of you are using the other book also, never mind, take down these questions and write down the definition. Proper definition. What is the first one? Indivision. Proper definitions you should know. You should write. We've already finished discussing all these topics. Osmosis. 4th is what? Osmotic pressure, is it? Osmotic pressure. And what is the other one? Active transport. Write down these 5 definitions, all of you, in the notebook. Write it down in the notebook, all of you.
Absorption chapter is finished.
No. We started the other day. You remained absent for 1 day or 2 days? Since you are saying. Genetics we are done.
I was not there for the complete introduction.
Complete chapter genetics you are not there. Super. And
This.
absorption the starting part of it now. Where you went?
School.
You all were having school?
Yeah.
But you were there no, the other day?
Yes. Yeah.
I am in
Okay, you are only one gold press, no? Holidays started or not? Holidays started or they're not giving you holidays? 3rd of June. Leave that school immediately. Why school has holidays they give? So which chapters they are in now?
Nervous system.
What are the chapters over?
Circulatory system, expiratory system, and
Three they have. This the plant physiology they have not done.
no.
Okay. So that means they'll be testing on this chapter also first, no? So after absorption, then we'll have to see which other chapters to start because you will have these chapters for your exam also, first exam. When is your first exam supposed to be? July?
September.
September. Okay, then we have to time. And still we have to then start with celebrations. What is the definition for the first one?
The phenomena by which
One at a time. Imbibition. That's the first definition. All of you are aware? What to write? What will you write? Imbibition.
The absorption of water by solid particles of a substance in their dry or semi-dry state without causing.
Okay, but it is also happening in the living or dead cells. That also has to be mentioned. So that also has to be mentioned. By the process of surface attraction, that also has to be mentioned. Second definition, diffusion.
Diffusion is the free movement of molecules of substance from a region of high concentration to a region of lower concentration when the two are in direct contact.
The molecules are in direct contact.
I mean, article is given as moment in easy words. Movement of substance from higher to lower concentration.
When molecules are in direct contact, you have to write down the complete answer. When the molecules are moving from a region of high concentration to a region of low concentration when the molecules are in direct contact. If they are separated, then it is osmosis. When they are in direct contact, then only it is just diffusion. all diffusion, all methods of diffusion, or when I talk about diffusion and osmosis, what is the basic difference? Osmosis is a form of diffusion. The diffusion, all forms of diffusion are not osmosis. So that you'll have to remember. Done? What about the next, osmotic pressure? So that was the last topic that we were discussing the other day. I'll continue again from this. But what was the definition that we had learned the other day?
It is a minimum pressure that must be exerted to prevent the flow of the water molecules when both the solutions are separated.
Allowed to prevent the flow of water molecules or solvent molecules into a solution. When?
And they are separated by a certain
membrane. Okay. And let us finish with the definitions first. Active transport. What was the concept of active transport definition? What will you write?
From a region of lower concentration.
From a region what? That is how, that's why I asked you to write. Write it proper way. Once you write proper way, then you understand that there's a particular format in which you write. Active transport, how will you define? Again, you are starting your answer with higher concentration, lower concentration. What is active transport? Define active transport. What will you write? Molecules of what? Water? Solute. Movement of solute. Movement of molecules of solute from a region of lower concentration to a region of higher concentration using the through living cell membrane using energy molecules in the form of ATP. Right? Complete answers. All of you make a habit of writing complete answers. See that whatever the question demands, the basic key terms, the format, you have to clarify. You have to write it clear. Otherwise, there are chances always for deduction of marks. Let us now continue with this concept. The first thing that, again, I want to start with is a concept of active transport. Now, osmotic pressure. So the other day we had already talked about the concepts of osmosis. And now, a little bit more again, this was the last topic that we were discussing the other day. Let us now again try understanding the concept of osmotic pressure. Anybody has a doubt? Please clarify. So we were talking about osmotic pressure, and then we said that this is a solution. Same thistle funnel we are taking, and we are taking a solution inside, and the thistle funnel is fastened with the semi-permeable membrane, same like the previous setup that we are talking about. And the difference here in the case of osmotic pressure, an activity to show osmotic pressure is, we are only talking about a weight that we have, a piston that we have used, and certain weights that we have put over the piston. Only that is the difference here, which we were not taking in the previous experiments with osmosis. What did I explain you the other day? That when I talk about the concept of osmotic pressure and the regular concept of osmosis, what is the difference? Though this is water and though this is the sugar solution, this concept remains exactly the same. Again, I repeat. So, though there is a concentration difference on either side of the semi-permeable membrane, there is a concentration difference on either side of the semi-permeable membrane. And if this is so, the other day always we had learned the movement of the water is supposed to be this way. The movement of the water is supposed to be always this way. The movement of the water is always from a dilute to a concentrated medium. But now in this situation, with the use of the piston and the weight, the movement of the water molecule could not happen. How do we understand? After waiting for a particular period of time, we get to see that the level of the solution is just as it is. The level of the solution does not change, and the level of the water also does not change. And they remain almost the same, with little bit of a variation, but not like the regular experiments on osmosis that we had said. Why now like this? It is because since there is a pressure exerted, since there is a pressure now on the semi-permeable membrane, let us consider semi-permeable membranes like the second department. Now, since there is a pressure applied, even though there is a concentration difference, the water molecules which were supposed to freely enter, the water molecules are getting a reverse pressure. The water molecules are getting a reverse pressure, so that's the reason they are not able to enter. This is what is responsible for keeping the level of the solution as well as the water in the beaker exactly the same even after the experimental period of time. So how do we conclude this? What is happening? Whenever there is a pressure on the reverse side, though there is a difference in the concentration, the movement of molecules from a solvent, this is a solvent medium, to the solution medium, which normally happens, now cannot happen. So are we not saying that this is the minimum pressure which can prevent the movement of solvent molecules into a solution? Yes. So this is the pressure which is not allowing this type of a movement, the movement of solvent to a solution, the movement of water to a solution. If this is the force and if this is the phenomena that we are talking about, if this is a pressure which can prevent the process of osmosis, we call it as osmotic pressure. We call it as osmotic pressure. But will this be the same when I'm talking about using some other membrane? Say suppose now I'm talking about an impermeable membrane. Can I say that that is also osmotic pressure? No, if it is impermeable membrane that we had learned, impermeable membrane was as it is not allowing water molecules to enter. It has nothing to do with the pressure. So the concept of osmotic pressure and the use of an impermeable membrane is not the same. In case of impermeable membranes, you need not apply any pressure to stop the process of osmosis. Just understand, just because of application of pressure, we have not changed anything as a membrane here, but still the movement of water molecule is stopped. The net movement has become almost zero. So this concept, this pressure which can minimize or almost prevent osmosis, we will call it as osmotic pressure. This also a little bit we had discussed the other day. Tonicity, again the definition of this word is important when we are talking about tonicity. The other day if you remember, what did we discuss was, I was talking about a cell and then I said that the condition of a cell, what would be the condition of a cell under which it will gain more water or it will lose more water. No? Those were certain conditions that we had studied. What were those three types of solutions we had studied the other day, which decides the behavior of a cell in that solution? So we had come across that if the cell is gaining too much water, if the cell is gaining too much water, what is that process called? What is that process called? Endoosmosis. When will a cell show endoosmosis? When the cell is in a hypotonic medium. When the cell is in a hypotonic medium. And now, again, if I'm talking about a cell which is losing too much of water, what is this process called? Exosmosis. Remember, this process would be exosmosis. And when will exosmosis happen? When a cell is in a hypertonic medium. And again, to remember, now that I'm talking about the definition of the hypertonic and the hypotonic solutions, how did we define these words? What is a hypertonic solution? What is hypertonic solution? Are we not comparing this solution with the cell? We are comparing, no? So with reference, now I may say that with reference to this solution, the cell may be dilute or the sap may be dilute. Outside solution is hypertonic, the sap is dilute. But that may be with reference to this solution. The moment I put this cell in some other solution, the conditions may be just the opposite also, no? So the moment I talk about isotonic and hypotonic and hypertonic, remember, we are actually talking about relative concentrations. We are talking about relative concentration. What is the meaning of relative? The last day also I told you, whenever you are writing down these definitions, you are actually comparing two different media. Just now as I told you that I am placing this cell in some 20% sugar solution, then I understand that this is 20% sugar solution is a dilute concentrated medium. This 20% sugar solution may be considered as a hypertonic medium for the cell. But I may immediately put this cell in some 5% sugar solution. Maybe at that time I may find that that 5% sugar solution is acting as a dilute medium for the cell, hypotonic. So now to understand that whenever we are talking about these words isotonic, hypotonic, hypertonic, we are talking about relative concentrations of two different medium, and based on the relative concentrations, if I say that the sap has got 25% sugar solution, this has got 20, is the direction of the movement of the water molecule remaining the same? I am saying that the concentration of the sap is now 25%. Concentration of the sap is 25% sugar solution. Try to understand what I am saying. Concentration of the sap is 25% sugar solution. The moment I say 25%, what are you supposed to understand? 100 parts water. I'm sorry, 25% means 75 parts water, 25 parts solute. 25% solution and ye ho gaya outside is 20% solution. The cell is in a 20% solution. Will the direction of movement of water remain same? No. Will the direction of movement of water remain same? No. What should the correction be then in this diagram? This is a dilute medium or this is a dilute medium? ये 20% है, ये 25% है. कौन सा dilute medium है? ये है dilute medium. यहाँ पे water ज़्यादा है, यहाँ पे water कम है. From where to where should water move? From here to here? From here to here? Or here to here? Which one is dilute, which one is concentrated? Now when I'm comparing this and this, I am talking about 25% here, 20% here. This is a hypertonic still. Now I have to write that this is a hypotonic medium. In comparison with the cell, now this becomes a hypotonic medium. If this is a hypotonic medium, the direction of the movement of the water changes absolutely. So earlier the situation was different. I said that the cell we have placed in a hypertonic medium. But now that I defined the concentrations, now immediately from the concentrations you are clear. Wherever there is a dilute medium, from the dilute to the concentrated, the water will always move. I said the moment you have to understand the concept of tonicity, you are understanding that tonicity means the relative concentrations of two different media. Will this decide the movement of the water molecules?
Yes.
Yes. So the relative concentration will always decide from where to where the water should move. The relative concentrations of two different media will always decide from where to where the water should move. Now, if I put here something around 45%, again, will the arrows remain the same? Tell me. Will the arrows remain same? Yes, absolutely. But if I put here 60, will the arrows remain still? Now, so every time we talk about differences in concentration, the movement of water molecule is changing. So that's why we are understanding that whenever I compare a cell, can I talk about that cell every time that the cell sap is hypertonic? Or can I say that the RBC cells are hypertonic? No. In what type of an environment, in what type of a solution it is kept? In what type of an environment it is, that will decide whether the cell is hypertonic or the external medium is hypertonic. The cell is hypotonic or the external medium is hypotonic. But anyways, the rule will always remain same. The water molecules will always flow from hypo to a hypertonic medium. So always to remember, the tonicity always will decide the direction of the flow of the water molecules. The tonicity will always decide direction of the flow of the water molecules. And what did we learn the other day? If the water molecule constantly fills in, if the water molecule constantly enters, if the process of endoosmosis takes in a cell for a longer period of time, then what may happen to this cell? The cell may burst. But that does not happen that very frequently in a cell, in a plant cell. Also, if I talk about aquatic plants, say suppose I'm talking about aquatic plants. Even in aquatic plants, the bursting out of the cells would be not happening so frequently. Otherwise, we talk about so many aquatic plants, and they are very much flourishing and growing because they have got the cell membrane also, the cell wall also, which can balance this. Now, with reference to this, we are going to understand turgidity and flaccidity once we are more or less aware about all these differences in the tonicity. Let me talk about a plant cell. Just now I said that a plant cell generally does not show the process of bursting. But the plant cell is always in a hypotonic medium. No? I talk about the lotuses, I talk about the water lily. They are always in a hypotonic medium. So if I understand the concept that the water flows from a hypotonic to a hypertonic and makes the cell swell up and makes the cell burst, then the plants in the water should never be flourishing. They always should be dying out. But does it happen? No, that does not happen. That means there must be some types of pressure or certain type of phenomena working there. So I may talk about a cell, a plant cell, and when we are talking about this plant cell, say suppose we first of all understand the process of endoosmosis happening. Now that the process of endoosmosis is happening, what is the first thing that would happen? After the process of endoosmosis, the volume of the cell sap increases. The volume of the cell sap is increasing and the volume also of the vacuole will increase, the space. The vacuole will increase in size. But immediately we cannot say that now that the volume has increased, now that the volume of the sap has increased and the vacuole has increased, the cell is on the verge of bursting out. Not exactly. Because this cell contents, whatever amount has increased, they will exert a pressure on the cell wall. The increased cell contents, they will exert a pressure on the cell wall, and if this pressure continues for a long time, then the cell will burst. If this pressure continues for a long time, yes, then the cell will burst. But immediately what happens? There will be a counteracting pressure that the cell wall acts on the cell contents. To counteract, to save the cell, the pressure which the cell contents are exerting on the cell wall, this will be counteracted by pressure of the cell wall on the cell contents. The pressure of the cell wall on the cell contents. The pressure of the cell wall on the cell contents, and this is these two counteracting forces which are always acting on plant cells, especially in the aquatic media. These are the two counteracting forces which are acting together in such a way and prevent the cells from damage, prevents the cells from bursting out. Now, what are these pressures? To remember their names, we are talking about the pressure which is, or the force which is acting, the force which is exerted by the cell contents of the cell wall. We will talk about this pressure as the turgor pressure. This is known as the turgor pressure. And the pressure exerted by the cell wall on the cell contents, this is known as the wall pressure. So both these, the turgor pressure and the wall pressure, are counteracting forces which prevent any particular cell from bursting out. But let us also understand two conditions known as turgid cell and flaccid cell. What is the meaning when I talk about a turgid and a flaccid cell? And what would happen if the flaccid condition continues for a long time? What does that actually lead to? Now, if I am talking about the turgid condition, I may understand that this is a regular plant cell and it has got the vacuole. Where is the cell placed? The cell is placed in water. What would happen after a particular period of time? After some particular period of time, we may understand that the cell must have undergone a whole lot of endoosmosis, and the cell is now increasing the content of the cytoplasm as well as the sap inside. The volume of the sap majorly has increased. The size of the vacuole has increased. This particular cell is now known to be turgid. This particular cell, which has shown an increase in the amount of the sap and the size of the vacuole, where if I talk about the cell wall also, I should say that in a turgid cell, the cell wall and the cell membrane, they are close in contact almost. The cell wall and the cell membrane, they are just pressed against each other because the vacuole is expanding. The amount of the sap is increasing. So when we are talking about a turgid cell has got adequate amount of solutes and large amount of water. But when will this turgid cell reverse the condition? When will the condition be reversed? If the turgid cell comes in contact with a hypertonic medium, when the condition would be dry or extremely concentrated. When the condition is dry or extremely concentrated, then what will be the condition of the cell? This cell will start undergoing certain changes. What would be the first change? The vacuole will start getting shrunken. The vacuole starts getting shrunken. How did the vacuole get shrunken? Because the vacuole or the cell has started giving away so much of water now. Why? Because the external environment of the cell has changed. What has happened? The external environment of the cell is no more a hypotonic medium. Rather, it must be a hypertonic or a dry medium. Hypertonic or a dry medium. What is this process called? Exoosmosis. So when we are talking about exoosmosis, now the cell wall and the cell membrane, they have started spacing out. The cytoplasm volume has started decreasing. The size of the vacuole has started decreasing. The amount of the sap has started decreasing. But if this continues, meaning if the cell is still in the hypotonic medium for a longer period of time, then what? The cell is on the verge of death. The cell is on the verge of death. So now, this cell, we will say that the cell is flaccid. The cell progressively has lost so much of its water. The size of the vacuole has decreased to a great extent as compared to this. So if this is a turgid cell, we have to compare this cell's naming that this is a flaccid cell then. How do we understand the turgid and the flaccid cell in a plant? Can we understand by looking into the leaves that the cells of the leaf must be turgid, the cells of the leaf must be flaccid? Can we understand? Yes, when you look into the leaves and we find that the leaves appear to be fresh and then appear to be quite shiny and looks waxy also in certain type of leaves. If I talk about the mango and all, they look quite waxy and shiny. Understood that the cells must be all turgid. But when the leaf appears wrinkled and dried out and wilted, then we understand that the cells must be in this particular condition. But if this continues for a longer period of time, then what? If this continues for a longer period of time, we will come across that the cell wall and the cell membrane have spaced out. What about the cytoplasm? Concentrated, because the cytoplasm has given away so much of water. What about the vacuole? The vacuole also has constricted. And in the adverse condition, we also come across that the cell wall also will start getting dried out. And this type of a condition is known as the plasmolysed condition of a cell, and the phenomena would be termed as plasmolysis. This would be termed as plasmolysis. So when does plasmolysis happen? When a cell is in the hypertonic medium for a long period of time, prolonged period of time. Because the cell has been constantly releasing away water, the cell is constantly undergoing the process of exosmosis. The cell wall, the vacuole is almost squeezed out, the vacuole is almost dried out. What about the cytoplasm? The cytoplasm has given away most of its water. The cytoplasmic contents are all concentrated. The space between the cell wall and the cell membrane is also filled with some concentrated solutes because the water part of the cell is almost lost. This is a plasmolysed cell. If continued for a short period of time, the cell may die out. But immediately, when I talk about that these plant cells, when they are quite dried up, sometimes we get to see, let's say suppose, sometimes when we come back from a holiday of 5, 6 days, 7 days, and we get to see some of our potted plants quite dried out. No? We put some water, and then we get to see next morning again they are growing fresh. But sometimes they remain dried out and they never revive back. So if these cells are not completely dead, instead of the hypotonic medium, what can we do is place them in a dilute medium again, or try to make their surrounding hypotonic again. So if I talk about the potted plants which we have come back from a holiday and seen that the leaves are all drying out, so what do? Immediately put some water, trying to make the surrounding of the cells hypotonic. Dilute, trying to make the surrounding of the cells hypotonic. Then immediately what should happen? The cells are supposed to revive, provided they have not died out. Then if they have not died out, yes, the cell membrane and the cell wall will take their regular position slowly. And what would happen? The vacuole will get back to its regular size and the adequate amount of the cell sap will fill in. And if that is happening, this is what we are talking about as deplasmolysis, and the phenomena known as deplasmolysis, and the cell known as deplasmolysed. The cell is now known as deplasmolysed. It's a revived cell now. This cell has revived from the plasmolysed condition. So how can a plasmolysed cell be revived back? By making the environment hypotonic. So one of the best way would be adding so much of water to the environment. So here we are talking about certain terms in relation. What are some of the terms you understand one after the other? We talk about what is a turgid cell. What is a turgid cell? And then what is a flaccid? So how will you define these two terms now? A cell which has adequate amount of water in the sap and which is tight filled with water, that is a turgid cell. What is a flaccid cell? A cell which has given away large amount of water. A cell which has given away large amount of water by the process of exosmosis is a flaccid cell. Then what is a plasmolized cell? A cell which has lost tremendous amount of water and the vacuole has shrunken, constricted, cytoplasm is extremely concentrated, and the space between the cell wall and the cell membrane is filled with concentrated substances, solutes. So you get this question that what will happen in a plasmolized cell? What is between the cell wall and the cell membrane in a plasmolized cell? There is large amount of concentrated glucose molecules and concentrated solutes basically, because they are no more in the soluble form, because the water part is all gone, the water part is all going out. So what are the terms in a relation? Try to understand what is turgid. The condition will be known as turgidity. This is a flaccid cell. The condition will be known as flaccidity. This is a plasmolized cell. The condition is known as plasmolysis. And that is a deplasmolized cell. The condition is known as deplasmolysis. Okay. Now, when we are talking about the concept of plasmolysis, remember, when we are talking about plasmolysis, there are some applications of plasmolysis also that we can think of. Give reasons, such and such questions you get, many of them. Now, when we are talking about plasmolysis, say suppose I talk about one example of plasmolysis. What could happen to the root cells if in certain agricultural lands so much of fertilizers fertilizers are sprayed in excess? What will happen? What are you expecting? That the root cells in a plant, when fertilizers are sprayed too much in excess, what should happen to those plants? What should happen to those root cells? The root cells will start dying out. The roots will start dying out. Why will the roots start dying out? Because they will all suffer from plasmolysis. The root cells will all start suffering from plasmolysis because the external environment of a root normally is supposed to be hypotonic or hypertonic. Hypotonic. Normally I told you. तो अभी इतना सारा fertilizer देने के वजह से now the external environment is becoming hypertonic. So the root cells which were supposed to take water, now they are giving away water. So the root cells are in the process of dying out by plasmolysis. How can we revive the situation? By adding adequate water to the soil. By adding adequate water to the soil, the condition can be revived. Now, there are so many questions that you will get always to apply the concept of plasmolysis for those. Some we will do also now. Say suppose now all of you must have sometime or the other you always suffer from some throat infection, cough, etc. No? So during the process of throat infection, doctors always ask us to do gargling, no, saline gargling. Yes. Why? Do you know why? Saline gargling. We take that water, and in that water, say suppose if you take one small glass of water, we are supposed to add 1 spoon of salt. So in 1 glass of water, 1 spoon of salt, the solution is becoming hypertonic. That is quite, quite a lot. In 1 tumbler, 1 glass or 1 cup, 1 spoon, that is a normal amount to be added. That is hypertonic. And now that you are gargling, the bacteria which is there in your throat, which is giving you the infection and the pain, surrounding the bacteria, the environment you are making hypertonic. So each and every bacteria, they are losing excess amount of water, whatever amount of water they have in their cells. They are losing that water and they are dying out by plasmolysis. So these are certain examples that you'll have to remember. Whenever we get cut or wound and there's a bacterial infection, again the doctor asks us, why with saline water? All these concepts that we do, all these processes that we do is destruction of a cell, whether we talk about a plant or an animal, by the process of excessive exosmosis followed by plasmolysis. Excessive exosmosis will always follow plasmolysis. Look into page 44. Take those questions, see if you are able to answer. Progress check. Karo, fatafat. Progress check. There are many terms, definitions, and applications in this chapter. You have to go through. Page 44, I say progress check. See if you know all the answers of this question. In your book, in between, they don't have any questions. Now, you may try number 9. Those of you using the other book, 3.15. 3.15, you can take number 9, number 12. 3.15, number 9, number 12. The same concept. Number 9, number 12. Did you get the answers for all? Did you get the answers for all? You are using Selena? I asked you to do that progress check. Did you get that? You also know? Same book? No, you have a different book. No? Then why are you taking this book? So for the different book, I gave you different two more questions. Where's your book? No book. Why? You came for his birthday party today. You should bring your own textbook. On top of that, your book is a different one. I will ask them to solve questions from their book, and you will solve your book questions that are there in the book. On top of that, some common questions we will do. You take your book. Okay, now you do. From next class, you have to have your book, notebook, volume, everything you are supposed to carry. Volume questions we will do. Your own textbook you should get because you have a different textbook as it is. Now only for 2 subjects you are coming. Those 3 books you can carry, no? School you go with a huge bag. Did you get the answers, all of you? Any doubt? Which one?
1 second.
Osmosis includes diffusion, but not vice versa. The same thing. Just some few minutes ago, I told you the same concept. When I talk about osmosis, all forms of diffusion is not osmosis, but osmosis is diffusion. But what is the difference between osmosis and diffusion? Truly, osmosis only talks about movement of water molecules. It never talks about the movement of solute. Diffusion talks about the movement of all. Solute also, solvent also, gases also. So yes, since it's a movement from higher to lower, diffusion talks about osmosis. Diffusion includes osmosis. But all forms of diffusion is not osmosis again. So what is that statement telling me? The statement is saying, osmosis includes diffusion. Osmosis includes diffusion, but not vice versa. Osmosis includes diffusion. Osmosis does not include diffusion. Diffusion includes osmosis. Because osmosis, the definition of diffusion is movement of molecules of gas, solvent, liquids from a region of high to low. So osmosis is a form of diffusion. But when I say osmosis includes diffusion, no, osmosis does not include diffusion because osmosis only talks about water. Osmosis never talks about solid. Osmosis never talks about gases. So diffusion में तो gas और solids भी तो included है. Done? All of you got the answers? That's one. Done, all of you? Finished? Now we are going to study that what is the use of turgidity or what is the benefit of turgidity that the plant enjoys. Cells are turgid. Yes, in the plants, cells become turgid. Cells do become turgid. We have always studied. But there must be some benefit that the cell enjoys. There must be some benefit that the cell enjoys. So when we are talking about turgidity, let me just move. So when we are talking about the benefits, let us go one by one. What are some of the benefits that the plant cell enjoys? So when we are talking about the first benefit that the plant cell enjoys is stiffness to soft tissues. Stiffness to soft plant tissues. So when we are talking about stiffness to soft plant tissues, now what does that mean? When we are talking about because of turgidity, when we are talking about the leaves, say suppose for an example, when we are talking about the leaf cells are turgid. Only when the leaf cells are turgid, they are flat and spread out. And only when they are flat and spread out, the process of photosynthesis, absorption of sunlight or transpiration, all these processes can happen. When the cells are, when the plants, the leaves are, the leaves of a plant, when they are crumpled and wilted, will the processes like photosynthesis and transpiration happen at the same rate? No, not at all. So when we are talking about only because the cells are turgid or only when the cells are turgid, the leaf parts, the young green stem of a plant, and these are the soft tissues of the plant that we are talking about, which are the hard tissues of the plant. The stem, the branches, the twigs, the midrib, these are the hard tissues. We are not talking about those. We are talking about the soft parts of the plant. Unless and until the cells are turgid, quite a number of physiological functions can never happen. Quite a number of the physiological functions will not happen. Excuse me.
हो गया लिख के क्वेश्चन
तो आना ही नहीं आने
Now, next is we were talking about that turgidity now that we are talking about, turgidity would always relate to the stiffness of the plant tissues, and why that stiffness is required, so that the different processes can happen. Okay, so we were talking about primary two processes like photosynthesis, transpiration. No? Next is when we are talking about the other benefit. What would be the other benefit that the plant would enjoy? Let us talk about one after the other. The germination process. We would understand the process of germination. The process of germination. Now, can we relate the process of germination to turgidity of the cells? The process of germination to the process of turgidity that the plant cells would have. I may talk about that the seeds which are there, the seed cells are absorbing water. The seed cells are absorbing water. If the seed cells are absorbing water in an adequate amount, every cell is becoming turgid. Then only and this also partially a concept of imbibition also I have to remember, because the seed coat that we are talking about is a dead layer. Here, it's not only the concept of turgidity, but little bit of imbibition is also there. But majorly, let us talk about turgidity right now. Now, I talk about a seed which is in the soil. Now, this particular seed which is in the soil, it has to undergo the breakage of the seed coat. How will the breakage of the seed coat as it is happen? How does it happen as it is? Because this seed which is in the water constantly keeps on absorbing water, and now every cell of the seed, that's why we say that the process of germination will happen only when there is adequate amount of water in the soil, apart from the other two factors. Adequate amount of water also is necessary. Now, in our own homes also we get to see that the process of formation of the sprouts, the first thing that should happen is the break or the cracks that should happen in the seed coat. And for the process of formation of those breaks in the seed coat, the opening up of the tissues through the seed coat, that would very much be related with the intake of the water. Now that the intake of the water, yes, the seed has become a little bit bigger, and now that these type of tissues start emerging out, this will emerge out only when there is a breakage in the seed coat. The seed coat should break. When will the seed coat break? When the cells inside are so tight filled with water that they are exerting a pressure and the outer covering just tears off, breaks off. Now the emerging out of these tissues. But now in certain methods of germination, we also come across that these tissues, as these structures keep growing, they would keep moving above the soil and taking the entire seed above the soil. That's a method of germination. No, you have studied in the 9th standard. So when we are talking about that these process in which we also understand, yes, the entire cotyledon now can move up. The entire cotyledon now can move up. The seed can move up, and this is how the pattern of growth is. So how did this happen? How did the entire seed start moving above the ground, above the soil? There must be certain amount of pressure generated. What is the pressure? The pressure is only from the individual seeds, which were individual cells of the seeds which were so turgid. Every cell was so turgid, and that was generating a pressure, allowed the breaking up of the seed coat, and then the emerging up of the tissues, and then the coming above. Now that is what is mentioned in your book also as breaking through the hard ground. What is meant by breaking through the hard ground? Breaking through the hard ground means that they come up over the ground, over the layers of the soil, and then they start the process of growth. In this entire process, it is only the basic concept of turgidity that we are talking about, the major concept of turgidity that we are talking about. Next again, we will talk about the opening and closing of the stomata. Before I go to this manometer experiment, let us talk about the opening and closing of the stomata. Where did we talk about the concept of stomata? Did you come across the concept of stomata anywhere? Transpiration, photosynthesis. So now when I talk about the concept of transpiration or photosynthesis, again, let us talk about another. We talk about the opening and the closing of the stomata. Now, how is turgidity related to this? Again, we are only talking about turgidity. What are some of the benefits the plant enjoys? Now, when we are talking about a particular stomata, what would be the basic structure of the stomata? The basic structure of the stomata to understand the guard cells. Now, it has got the guard cells. And what is surrounding the guard cells? A whole lot of epidermal cells that it will have. A whole lot of epidermal cells that it will have. Now, surrounding the epidermal cells again, there are the xylem cells, not to forget. Let us start from here. Now, I'm saying that these guard cells, wider the stomatal opening, what happened? Itna pyar kyun aa raha hai us pe? Now, when we are talking about these guard cells, they have to become turgid. The more they are turgid, wider the stomatal apparatus. Now, this stomatal opening, which is known as the stoma, wider it is, the diffusion of inward diffusion of carbon dioxide is more, and the outward diffusion of water. Both processes we have to understand. Now, simply here we are trying to come across how turgidity may play a role. Turgidity, of course, will play a role because now that we talk about the movement of the water molecule from where? From the xylem cells. The xylem cells carrying the water from the soil, that is understood. Now, these xylem cells, which are in the veins and the midribs, very close to the guard cells, these xylem cells are giving water to the epidermal cells. These are the epidermal cells that I'm talking about. Epidermal cells. Now, if it is a condition where there's adequate amount of temperature, sunlight, dry environment as of now, what will happen? These guard cells, they are constantly absorbing water from the surrounding cells. Why? Because these guard cells, they are undergoing a process known as photosynthesis, remember. These guard cells, they also, since it is a plant cell, they also will have their vacuole. Since it is a plant cell, they also will be having the chloroplast. So now if I'm saying that they very well have the chloroplast and they are constantly undergoing photosynthesis, forming the glucose here in the sap, making the sap hypertonic. Because it is hypertonic, the water constantly enters. The water constantly enters the guard cells. I repeat what I told in a sequence to remember. I am saying that the guard cells are a typical plant cell. So they also should be having chloroplast. If they have chloroplast, I'm showing these organelles as chloroplast. If they have chloroplast, they will be also manufacturing glucose. Glucose getting dissolved in the sap. Sap becoming hypertonic. Surrounding cell, is it making glucose? But it is only getting water from the xylem. So which cell is having more water? This or this? Epidermal cell is having more water because this cell is utilizing water in the process of photosynthesis also, and gradually the sap is becoming hypertonic because of the addition of glucose. So every time this guard cell and this surrounding epidermal cell, when we compare, which is a hypotonic medium, this one. क्योंकि epidermal cell तो glucose नहीं बना रहा है. On top of that, it is not using the water much, जो ये cell कर रहा है. So now this particular cell is always running short of water. So if that is true, then this cell will always take water from the surrounding cell. Endoosmosis ki exoosmosis? Endoosmosis. With repeated endoosmosis, what is happening? This cell is becoming bulged out. The vacuole is becoming big enough. So much of water has filled in. What about the guard cell? No, the guard cell also will become, जो ये regular size ऐसे था. Now this guard cell also will not remain like this. The guard cell also will become extremely curved, and the guard cell also, the walls, the inner and the outer wall, the outer wall will curve in such a way that the inner wall is also pushed outwards like this. So if it is happening for both, so what is happening? The guard cell is becoming curved, and this is what is happening. The opening of the guard cell has widened up than before, because inner membrane will get pushed outwards and outer membrane पूरा curved हो जाता है whenever we are talking about the guard cell becoming turgid. Then what is the benefit the guard cell or the plant cells are enjoying? The plant cells are enjoying two benefits here. एक तो है inward diffusion of carbon dioxide and दूसरा the outward diffusion of water. The plants can give away their excess water and can take in a whole lot of carbon dioxide to carry on the process of photosynthesis. So what did we understand? What are the first three benefits that the plant enjoys? Because of turgidity, it can keep the plant parts stiff to carry out many processes. It can allow the process of germination and growth. जब तक turgidity नहीं होगा, as long as the cells, if the plant cells are not turgid, seed cells are not turgid, process of germination cannot happen. The stomata, the cells of the stomata should be turgid, then only the opening up of the stomata, and then only the diffusion of carbon dioxide and water. Now we talk about something different to try understanding that how does this process of turgidity also help in the movement of the water upwards. And this we will learn with the help of a apparatus. This particular apparatus that you get to see is known as the manometer experiment. This is also known as the manometer experiment. Can you see there? There's a setup there in your book also shown. What is happening here? There was a potted plant here. All of you can see this. There was a potted plant here. There was a potted plant here. So what they have done is cut out the upper portion of the plant, keeping a little bit of the stem attached to the soil. The length of the stem also should be definite. Minimum 3 meters should be there. So what they have done, cut out the upper part of the shoot, keeping a small portion of the stem attached to the soil. Now what they have done immediately is, to this cut out portion of the stem, attach a U-shaped or a bent glass tube. This is called a bent glass tube. So what they have done, after cutting, immediately this has to be done. After cutting, attach a bent glass tube, but the attachment should be very tight. That's why they have fastened it with some plastic or fastened it with some tissue tapes, rubber bands, or some plastic sheets. So this or some rubber sheets. So it has to be fastened properly, so that the stem and the glass tube, they should not be placed this way. If this is the stem and this is the glass tube, they should be fit exactly this way. They should not be dislocated this way, then we will not understand the experiment at all. So it should be exactly like this, okay? So now it is done. Next what? Before the shoot is cut out, also to make sure that the soil is well watered. The soil has to be well watered. Already in the bent glass tube, a particular amount of mercury is taken. Already in the bent glass tube, a particular amount of mercury is taken and the level of the mercury is also checked. This is kept for a short period of time, for a few hours, for some 4 to 6 hours, under normal temperature, under sunlight. It is kept. Now what is observed is, after some 6 to 8 hours, what do we get to see is that some amount of liquid is filling up here in this tube. Earlier, there was only mercury. So earlier in this tube, mercury was filled up here. Earlier in this tube, mercury was filled up here. Now, after these 4 to 6 hours of time, what has been observed is, what has been found is, that the mercury constantly keeps on moving, and it has taken a position here. And this entire region is filled with a liquid. Now, from where did that liquid come? From where did that water mixed with some salts perhaps? From where did this liquid come? It is understood that this liquid must have been released from the cutout region of the stem. This liquid must have been released from the cutout region of the stem. So whenever we are talking about the cutout portion of the stem, now if we are talking about the potted plant, this stem that we cut out, in an oblate, there are a whole lot of cells in the stem. So obviously the stem would be releasing, or the stem would be conducting some amount of liquid. Which tissues must be involved in the conduction of the liquid? Xylem. So now, whether we are cutting down, though we are cutting down now the upper portion of the shoot, the process of conduction will not stop. The process of conduction will still continue. So because of the process of conduction of xylem, which is still continued, because the soil is well watered, that has to be taken care. So what is happening? The xylem is still continuing the process of conduction of the water, and that is the sap which is getting filled up in that tube. That is the sap which is getting filled up in the tube, and the level of mercury was here, from here to here. The initial level of mercury was here. Now the level of mercury is here. Now the position of the mercury in the tube has changed. The mercury has changed its position. पहले यहाँ से यहाँ तक था, अभी धीरे-धीरे move होते, now it has come to this particular position. And will it still gradually keep on moving? Till when will it keep moving? It will keep moving as long as there is adequate amount of water in the soil. So, from this particular activity, we are not only talking about a concept of turgidity, we are understanding 2-3 concepts here. पहले तो ये that whenever I talk about now the cells of the xylem, I want to understand the cells of the xylem. Say suppose I talk about that these are the plant cells. This particular plant cell has taken in water. This particular plant cells have taken in water. Once this particular plant cell has filled water, once this particular plant cell has become turgid, how will it behave? Will it just burst out? No, it will not burst out. It will give its excess water to this cell, and again after some time take more water. This cell, when it has reached a condition, something to be termed as turgid condition, when this cell has reached its turgidity state, it will not burst. What are the two counteracting forces which prevent the bursting of a cell? Turgor pressure and wall pressure, the two counteracting. So now that I talk about the cells of the xylem, how are they behaving? These cells, when they are turgid, full of water, the water will move to the next cell. Again the same thing, it is now filled with water, turgid condition, the water will move to the next cell. What is this process known as? This process is known as cell-to-cell osmosis. This process is known as cell-to-cell osmosis, which is responsible for the movement of the water in the xylem in tall trees. So only the concept of osmosis that we have seen at the level of the root cells, the root hair cells, is only because of the hypertonic condition of the cell sap. But because of that osmosis or hypertonic condition of the cell sap, is that hypertonicity good enough to allow the movement of the water happen all throughout the stem and the branches? No. For that, some other different factors and forces and pressures are required. So here, relation with turgidity, we are saying every cell becomes turgid, gives away water to the next, gives away water to the next. This is how the water keeps on moving all throughout the xylem to reach the cells of the leaves in a plant even 50 to 100 meters high. So when we are talking about this particular benefit, yes, the plant enjoys this particular benefit because the turgidity is allowing cell-to-cell osmosis and the movement of the water to the upper parts of the plant. Then the last that we are talking about is turgor movement. So this was the fourth, and the fifth that I talk about would be the turgor movements. Now let us talk about turgor movements. Turgor from the word turgor, pressure, turgidity. So what are the four benefits? First of all, we had understood. The first is stiffness, second is germination, third is opening, closing of the stomata, and fifth is fourth is cell-to-cell osmosis, allowing the movement of the water minerals to the upper parts of the plant by the way of xylem. Now we are taking up a different concept, of course related with turgidity, but we are talking about a different concept, that is turgor movements. What is turgor movements? Let us try to understand what is known as turgor movements here. A condition something like this, I think all of you had come across. No? This is a touch me not. What happens here? What happens here? If we touch the lead, the entire row of the leaflets closes down. Does it revive on its own? Does it revive on its own? Within a short period of time, it would revive on its own. What happens? Is there a relation of turgidity here also? Yes. There's a relation of turgidity here also. Let us see. First of all, when we are talking about these type of plants, what is happening here? When I talk about this type of plants, I may understand that these type of plants are something like this, where there is a row of leaflets, where I may understand that this is one particular leaf, I understand this is one particular leaf, I understand this is one particular leaf. No? So there's a row of leaflets like this, and the entire leaf which is divided into small leaflets like this. Exactly the same thing that I was showing you. This is one leaf, this is another. Now, as the water is moving up, why? What is the reason that the leaves close? What is the reason that the leaves close? Now, when I'm talking about that the water is rising up, the water is rising up, and then the region where the stem meets the twig, which will carry all the leaves, this plays an important role. We'll come across how. So first of all, I'm talking about that these leaves are all having an adequate amount of water. They are all in their turgid state. What has happened? Maybe you have touched here. Maybe you have touched here. This is acting as a stimulus. This is acting as a stimulus. Now, in the concept of nerves also, somewhere or the other, always in the 7th, 8th, 9th standard, always you have come across the word stimulus. What can we define the word stimulus suitable to this concept right now? I would say stimulus can be any particular agent which can bring about a disturbance. So yes, I'm talking about a disturbance. Now, is it only that when we touch, there's a change and the closure of the leaflets? Or it can be because of a lizard, it can be because of a butterfly, it can be because of a bird also? Yes, it can be because of anything. Whenever there's a disturbance, that is acting as a stimulus. So now, when there is a stimulus, all the cells of the leaf are turgid. Turgid. हर एक सेल के अंदर बहुत सारा water already filled है. And I already told you in one of these explanations that what would happen when one particular cell is already turgid. How will this cell behave? Give water to the next cell. One cell is already turgid and that cell is little bit disturbed, the water will immediately rush to the next cell. That is what is happening here. That is what is happening here. So now, initially, if I understand that this was the stem, and there's a stimulus that we apply, what has happened immediately? The water from every cell starts moving to the next cell. The water from every cell starts moving to the next cell. So now the water movement has happened, the water movement has happened, the water movement happens in all directions. This leaf, a small leaf, where there may be 50 to 100 cells, and every cell has given away water to the next, given away water to the next. As a result, the water is all rushing out, the water is all rushing out, the water is all rushing out this way. So from every particular leaf, from every particular cell, since the water is, or since the cells are disturbed, the water molecule changes its position. And the water molecule starts moving away, starts moving away, starts moving away, and respectively, these the cells of these leaves are becoming flaccid. So for every single leaf, if I see that yes, this particular leaf is giving away water to the next region, this is giving out water to the next region, this is giving out water to the next region. So what is happening to this? This in turn becoming flaccid. They are losing their turgidity. So also this. So progressively, there's a chain of movement of water happening, one on top of the other. These cells are all giving away their water. These cells are all giving away their water. And then ultimately, in the form of a circulation, what will happen? The water starts accumulating at one particular region and making the base of the stem or the twig and the stem little bit swollen like this. And when it is swollen, this particular region is known as the pulvinus. When it is swollen, this is what is known as the pulvinus. So what I told you right now is that these leaf cells, when they are flat and spread out, just like I was talking about stiffness of the soft plant parts, no stiffness of the plant tissues, I told you. For the stiffness of the plant tissues, leaf cells should have adequate amount of water. Leaf cells should be turgid. Yes, that is what is happening right now. I'm telling you that when they are open up, are the cells turgid? Yes, all of them are turgid. When they are open up, when they are spread up, every cell is turgid. But the moment there is a stimulus, the water molecules, in a haste, they start moving from one cell to the other, one cell to the other, one cell to the other. That is what happens. As a result, every single leaf runs short of water because so much of water molecule has started moving away from their cells to the next and the next and the next regions of the twig, entire twig. So this is what will happen. If you see here, this is what will happen. And as a result, these leaves, which has already suffered from a change in the water molecule position, they have started suffering from a flaccid condition now. Every cell turns to be flaccid, resulting in a complete closure that we understand, resulting in a complete closure. But will this remain forever? No. After some time, these water molecules again will start circling and moving back because the water molecules are not losing anyway. Can I say that the water molecules are getting lost by the process of transpiration or anything similar? No, the water molecule is just circulating one from one part to the other and then coming back to the stem and then again moving back. So something like circulation ho raha hai in the water molecules of all these parts from one part to the other. Pehle flaccidity develop hota hai. But the water is accumulated again in the stem and in this part. Then from here again the distribution will start. So from here when the distribution starts this way, the revival also starts again. So yes, tell me.
Why does it happen in other plants?
Why does it?
Why does it not happen in other plants?
Because when we are talking about, now it will happen in this type of plants and some related type of plants which has got very few or weak arrangements of the xylem, or these are comparatively small herbs. This will not happen in these big trees at all because they have got elaborate system of xylem tubes, so many veins and the midrib. But these type of plants, these type of leaves, they don't have first of all anything known as the midrib and all. The midribs are always supposed to be the base support for these type of leaves, but they don't have anything like this. So they have basically one particular point which joins the twig with the stem. That is their support. That's the main tissue. So first of all, they will lose water. From turgid, they become flaccid. Then from flaccid conditions, again they will become turgid, and that is when they will open up again. So here, are we talking about the utility or the application of turgidity to the plant parts when we are talking about mimosa? Of course, when we are talking about this type of flower, when we are talking about this type of leaves which are sensitive, that's the same thing happening. Again, when I talk about certain more examples, something like the pitcher plants, when I talk about the Venus flytrap, there also what is happening in these type of plants, some insects come in contact, or etc., etc. What happens? For the pitcher plant, the upper arrangement of the leaf would just fall on the mouth-like structure, and the entire structure is closed. I talk about the Venus flytrap. Again, when the insects keep meandering, they would close. This is only because of the loss of turgidity that the cells are suffering. Because the moment there is a stimulus, the movement of the water molecules, first of all, turning all the turgid cell flaccid, and since there is no water loss, वो flaccid condition फिर से revive हो जाता है. Flaccid again will be converted back to turgid conditions. Okay, so they revive back. Take a break. We'll have to continue again after some time.
चप्पल उतार के भैया आएगा
पानी तो एसी भी चालू नहीं है इधर का ऐसे मैंने तवे में रखा है
भाई वो तो भी एक विषय है क्या है अरे आगे इसकी कमी पूरी कर रहा है हम उधर मिले हैं भाई मैं को जाना है
मुझे अपने घर जाना है
और 130 मिनट है एग्जैक्ट
130
भाई 5 मिनट के लिए ब्रेक का भी
नहीं मैं 55 तक ब्रेक लूंगा मैं
बिल्कुल नहीं मालूम इसी टाइम तक जाना
मेरे आर्यन ट्यूब से लेके आ रहे हैं
ऐसा दे
ये हो गया मेरा
आयुषी आ रहे हैं तो उसकी टॉपिक थे कोई पूरी कर